Bagong pagkaaral, samahan yung nasira ng tiyan. SUPERCUT, dagdag kaalaman, makinig ka na sa aming gaswainan. SUPERCUT, nagkampara sa payan, samahan niyo si Pilar Tiyan. SUPERCUT, your daily source of science is now here. Halina't makinig sa SUPERCUT.
Sawa na ba kayong magbasa ng napakaraming articles para lang magka-idea sa inyong next research topic?
Or hirap na ba kayong maghanap ng model paper na sasakto sa thesis idea nyo?
Worse, naumay na ba kayong makahanap ng reference paper only to find out na may bayad ito?
Then guys, you have come to the right place.
Welcome back to SUPERCUT, where we bring you scientific papers and ideas like no other.
So without further ado, SUPERCUT to the chase.
Last episode, we talked about the infamous surgery of attaching a modified pig kidney to a brain-dead person. Nakakaloka. It is certainly controversial, especially among animal rights activists. Pero hindi rin maikakaila ang potential nito to become one of the main sources of organ transplant. And now, it's time to read the super chat for today. Let's hear it from our sender at pretty-angel123. Ang sabi niya, Dear Sir Reynard and Madam Shena, I am Pretty Angel and senior high school student po ako. Sa totoo lang, pagod na ako maghanap ng inspiration for my research paper kasi ginawang individual ng teacher namin. My interests include genetic engineering, gene recombination, and biotechnology. More specifically, gusto ko po sanang mag-edit ng genes ng bacteria. Kaso hindi pa po siya na-isolate. Sabi pa ng teacher ko, wala pa atang method for that. Babagsak na po ba ako? Sana po may ma-recommend po kayo sa akin, SUPERCUT.
Wow, Pretty Angel. Grabe yung problema mo, girl. But anyway, don't worry. Perfect na perfect. Kasi may alam kaming kakapublish lang na article about this.
Oo nga. Kanina ko lang nabasa yung tungkol sa species and site-specific genome editing in complex bacterial communities. Fortunately, best friend ko yung author. Kaya I invited her here to inspire us with her team's research. Please welcome my bestie, Dr. Jorlene.
Hello, Dr. Jorlene. Kumusta ka naman, lalo na ngayon at bagyo season na ulit dito sa Pilipinas?
Hello, Reynard and Shena. I'm alright naman. Safe lang kami dito ng family ko sa Manila. Hopefully, kayo rin safe and sound.
SUPERCUT to the chase, Doc. Pwede ba namin malaman ano yung research nyo at papaano ito ginagamit?
Yes, sure! Na-mention na kanina na ang title ng paper namin ay Species and Site-Specific Genome Editing in Complex Bacterial Communities. Basically, may dalawang bahagi ang methods nito. DNA editing all-in-one RNA-guided CRISPR-Cas transposase o DART systems at environmental transformation sequencing o ET-Seq.
Okay, wait. Medyo nag-loading ako nun, Doc. Technical terms na agad kasi tayo. Pwede mo bang ma-explain sa ating tagapakinig, especially nung mga hindi alam kung ano yung study nyo, kung ano ang mga meaning ng words na to, Doc?
Okay, first, yung ET-Seq ay unang ginagamit para makilala ang mga bahagi ng ating microbial community. This is done through the use of randomly integrating mobile genetic element that identifies which microorganisms are available for gene editing. Pagkatapos ng ET-Seq, DART system naman ang gagamitin sa pamamagitan ng pag-insert ng CRISPR-Cas transposase system sa genome ng microorganism. Lastly, it uses gRNA for its specificity in gene editing. Once the target gene is identified, the DART system cleaves the sequence just like the usual CRISPR-Cas systems.
Medyo gets ko na siya. So, basically, similar lang to sa CRISPR, pero in this case, hindi na kailangan ng bacterial isolation, no?
That is correct.
So, Dr. Ano ang kahalagahan ng methods na ito? Mas madali ba ito gamitin o mas mura?
Malaki ang significance ng method na ito lalo na sa microbiology. Gaya ng nasabi kanina, ang method na ito ay hindi na nangangailangan ng isolation of bacteria bago makapag-manipulate. Ang classical approach kasi required mag-isolate, so ayun, medyo limited tayo kasi majority ng microorganisms ay hindi pa na-isolate. Isa pang advantage ng method na ito ay maaari nating ma-explore ang genes involved in interactions between organisms. At dahil diretso na sa microbial communities, makikita agad natin ang wild-type physiology kumpara kung magkukulture pa tayo sa lab.
Ang interesting naman, Doc. So, wait, paano ito ginagawa?
Oo nga, can you run us through the methods, Doc?
Sige, sure. Fasten your seatbelts kasi medyo mag-rapcad ako dito. Okay? So first, ETSEC identifies genetically accessible bacteria to assess the ability of individual species within a microbial community to acquire and integrate exogenous DNA. After testing its accuracy and detection limits, we use ETSEC to compare insertion efficiencies in the 9-member synthetic soil community after mariner transposon delivery by either conjugation, electroporation, or natural transformation with no induction of competence.
Okay, Doc. So, correct me if I'm wrong, pero from what I understood, the final readout of ETSEC estimates the proportion of each organism's population that harbored transposon insertions at the time the DNA was extracted, which is a combined metric of delivery, insertion efficiency, and mutant survival after delivery. Tama ba, Doc?
Yes, tama. Plus, we also identified preferred delivery methods to produce insertions in certain members. Okay, secondly, I'll talk about the targeted genome editing with CRISPR-associated transposases. We reasoned that RNA-guided CRISPR-Cas9 transposases could provide the ability to both ablate the function of targeted genes and deliver customized genetic cargo in organisms shown to be genetically tractable by ETSEC 12 and 7 to 19. Also, since ETSEC identified conjugation and electroporation as broadly effective delivery approaches in the tested communities, we designed and constructed all-in-one conjugative suicide versions of these Cas9 vectors that could be used for delivery by either strategy.
Earlier, you mentioned the DART system. May I ask how this works?
Well, these DART systems are comparable to those in the integrate systems developed in Parallel 12, but are barcoded and compatible with the same sequencing methods used for ETSEC.
Wait, Doc. Tama ba narinig ko? Barcodes?
Yes, tama. Barcodes. Basically, they are present on all transposomes used in this paper, which allows for the detection and the tracking of uniquely edited cells. Thus, DART can be used seamlessly with ETSEC for rapid assay of the efficacy of CRISPR-Cas guided transposition into the genome of a target organism in the absence of the selectable markers.
How about the targeted species and locus-specific community editing?
Okay, so medyo mahaba tong tinatanong mo ha. Give me time. Okay, wait. We reasoned that RNA program transposition could be deployed for targeted editing of a species within a microbial consortium. As an abundant member shown by ETSEC to be tractable by conjugation, we first targeted K. michiganensis. Conjugation was used to introduce the VC DART vector into the community with multiplex gRNAs specific to 2 locations in the K. michiganensis genome.
Wow, okay. So, you're really not kidding, Doc, when you said na mahaba yung process. I'm totally out of words and super amazed ako. Pero Doc, last question. If I may ask, ano yung delivery methods niyo?
Plates were incubated at the preferred temperature for the recipient community or isolate for 12 hours. Before growth was scraped off of the filter into the medium of the recipient community or isolate for downstream analysis.
And that's a wrap. Thank you, Bestie, sa pagsagot sa aming mga tanong. I have to cut our conversation kasi may important appointment pa raw si Dr. Hanggang sa muli, Doc.
Wala ka naman, Bestie Shena and Reynard. Thank you for inviting me and sana marami tayong na-inspire na tao sa araw na ito.
So, ayun nga, guys. Na-explain na sa atin ni Doc Jor yung mechanism behind the study. Pero, I'm going to ask you this. Shena, do you think na magagamit natin siya as a beneficial tool to better our society? And what are your thoughts regarding the benefits and risks of editing microorganisms?
For sure, may benefits na pwedeng makuha from this. From a broader perspective, like gene editing in general, it has risks. Pero, syempre, we also have the opportunity to use it for a multitude of improvements. Are you familiar with bioremediation?
Narinig ko na siya, pero sige, walk me through it.
Alam mo ba recently, studies have been focusing on using microorganisms to clean up areas that have toxic elements or unwanted chemicals. By that I mean, mga sites na nagkaroon ng chemical spill or naka-experience ng irresponsible waste management. Scientists today are already looking into different organisms to counteract the damage we've been doing to this planet. With this kind of ability, yung ability to identify strains and genes within a complex community, it makes things easier for fields of research like bioremediation. Kasi it brings forth an option that would allow them to further look for possible agents. After all, unintended consequences and unforeseen interactions after the release of GMOs is always a risk.
I see. O nga no, I mean, the possibility of creating organisms that may pass on advantageous genes to a different species in the environment that the GMO is introduced in is always present. Sabi nga nila, the risk of horizontal gene transfer is something that scientists should always be careful of when doing this sort of research.
I agree. Pero ikaw ba, Reynard? Other than the broad overview of the possible risk of gene editing, ano sa tingin mo magiging benefit ng study na to?
Good question. Wait. So, I feel like marami talagang benefits kahit itong singular study pa lang na inexplain sa atin ni Doc Jor. For example, in terms of understanding microbial communities, we'd be able to analyze gene fitness and function straight from the source, diba? Thanks to VC-DART editing and ET-sec detection. Hindi na natin kailangan mag-sequence ng paulit-ulit or gumawa pa ng complex processes just to determine kung may value yung genes present within the community. Moreover, ET-sec would allow for identification of genetically tractable bacteria within the microbial community.
Oo. Tapos it makes editing easier, di ba? By changing only a portion of the gRNA sequence in the VC-DART vector, traits of interest can be enriched within the community or isolated, if needed, kahit ang daming iba-ibang species na nag-i-interact. Not to mention DART can be used seamlessly with ET-sec for rapid assay of the efficacy of CRISPR-Cas guided transposition into the genome of a target organism in the absence of selectable markers. Ang laking bagay nun since alam natin na yung presence or absence lang ng ibang genes ang nagdedetermine kung magiging pathogenic or commensal yung bacteria. Then it's possible to find out quickly.
Exactly, Shanna. Huwag din natin kalimutan yung benefit na we can identify individuals other than by identifying genetically tractable strains. Pwede rin tayo mag-identify by locus-specific or site-specific genome editing. This gives a better overview, malalaman natin kung ano yung mga species na andoon. What's more is pwede nating silang i-manipulate without isolation. Ang laking bagay nun in terms of, the application of research. Crazy nga talaga. Mas mabilis pa sa fast gumalaw yung scientific advancement natin regarding things like this, no? Hopefully, a lot of other studies can capitalize on this. Alright, so supercut to the hard questions.
Oh wow! Ang bilis! Narito na tayo sa segment kung saan pag-uusapan namin ni Reynard ang ethical aspect ng research for today. So, first question, unethical ba ang genetic engineering at biotechnology?
Alright, another good question. So, unahan ko ng mga Spider-Man fans siya na. Ako, I've seen the new Spider-Man. Na kayo ba, guys? Pero anyway, ang sabi doon, diba, with great power comes with great responsibility. So, as scientists, we are expected to inflict no harm. Again, we should not inflict harm. Hindi lang naman sa biotechnology ang mapanganib, diba? Kahit sa pag-handle ng chemicals, nakakasama rin kung hindi tayo susunod sa proper protocols. So, nasa research practice pa rin talaga ang bottom line ng lahat.
Ito na ang favorite part ko. Sige nga. Are you for or against the application of such study?
Alam mo, I'm all for the progress of science. So, we can always improve our systems kasi and technologies. While at the same time, be careful lang talaga sa maaaring effects nito. Just like any other method that arise in this technological advancement, follow lang talaga sa proper procedures and proper protocols na prepared by the ethics board.
Dapat lang din talaga accountable ang mga people behind this method in case may damage control na kailangan in the future.
It's another day for science, Shanna, diba? Do you agree?
Indeed, Reynard. Kaya samahan niyo ulit kami next time sa talk show tungkol sa agham.
At para sa bayan, muli, ito po ang inyong Superman, Reynard.
Ito naman ang inyong superwoman, Shanna. At nakikinig kayo sa SUPERCUT.